This video is presented by Subi, lecturer from Emmanuel Arasar College of Pharmacy. Today we have see about nephron. Introduction. Nephron is the basic structural and function unit of kidney which helps in formation of urine. The word nephron is derived from the Greek word nephros, meaning kidney. There are about millions of nephrons in each human body. Human kidney. Types of nephron. There are two types of nephron on the basis of location. Region nephron. It is divided into cortical found in cortex, juxtamedullary found in medulla region. First we are see about the cortical nephron. These are the nephrons present within the cortex. These are short and is about 80% of the total nephrons. Here, vasa recta is not present. Next move on, juxtamedullary nephrons. These extended into the medulla. These are about 20%. Here, vasa recta is present. The vasa recta, the capillary networks that supply blood to the medulla. Vasa means vessels and recta means erect or straight. Means straight blood vessels or blood vessels that go straight with the renal medulla. Anatomical divisions of nephron. Renal corpuscles, glomerulus and Bowman capsules. Renal tubule. PCT means proximal convoluted tubule, loop of Henle. Descending and ascending DCT. Next we are see about anatomy of nephron. It is a long tube-like structure. Its length varies from 35 to 55 mm long. The structure of nephron comprises two major portions. They are renal corpuscles and renal tubule. At one end, the tube is closed, folded and expanded into a cup-like structure called the Bowman's capsule or renal corpuscular capsule, which has cluster of blood vessels called the glomerulus. These capsules and glomerulus together constitute the renal corpuscles. This is the structure of nephron. Renal corpuscles. It consists of glomerulus surrounded by a Bowman's capsule. The glomerulus arises from an afferent arteriole and empties into an efferent arteriole. The Bowman's capsule is made up of simple squamous epithelial cells and divided into three layers: outer layer, middle layer and inner layer. Outer layer. It is otherwise called as parietal layer. It is made up of epithelial cells with minute pores of diameter 12 nanometer. Middle, basement membrane. It is made up of fibrils, etc. This layer is selectively permeable. Inner, visceral layer. It consists of large cells called podocytes which bear finger-like projection called podocell. Podocytes are covering of glomerulus. It contains filtration slits which prevents the filtration of medium-sized proteins. Renal tubule. Next we are say about renal tubule. The renal tubule is a long and convoluted structure that emerges from the glomerulus and can be divided into three parts based on function. The proximal convoluted tubule, due its proximity to the glomerulus, this stays in the renal cortex. Loop of Henle or nephritic loop because it forms a loop with the descending and ascending limbs that goes through the renal medulla. The third part of the renal tubule is called the distal convoluted tubule, and this part is also restricted to the renal cortex. Bowman capsules extend to form highly coiled tubules like PCT. PCT continues to form the loop of Henle which ascends to DCT, which in turn opens into the collecting duct. Next we are see about proximal convoluted tubule. The blood brought by the renal artery is filtered by the glomerulus and then passed to the PCT. Maximum reabsorption takes place in PCT of the nephron. PCT is the region of renal tubule where reabsorption of essential substances like glucose, proteins, amino acids, a major portion of electrolytes and water take place. It is made up of simple cuboidal epithelium in them. It selectively secretes ions such as hydrogen, ammonia and potassium into the filtrate and absorb bicarbonate from it. Thus PCT maintains the electrolyte and acid-base balance of body. Next we are see about Henle's loop. Henle's loop has a descending and an ascending limb. Both the descending and ascending limbs show different permeability. The descending limb is permeable to water but impermeable to an electrolyte, while the ascending limb is permeable to electrolytes but impermeable to water, since the electrolytes and get reabsorbed at the ascending loop of Henle. The filtrate gets diluted as it moves towards the ascending limb. Henle's loop has a descending and ascending limb. Both descending and ascending limbs show different permeability. The descending limb is permeable to water but impermeable to an electrolyte, while the ascending limb is permeable to electrolytes but impermeable to water. Next we are see about DCT, distal convoluted tubule. The DCT, which is the last part of the nephron, connects and empties its contents into collecting ducts that line the medullary premix. The collecting ducts emit contents from multiple nephrons and fuse together as they enter the papillae of the renal medulla. Similar to PCT, DCT also secretes ions such as hydrogen, potassium and ammonia into the filtrate, while reabsorbing the bicarbonate from the filtrate. Conditional reabsorption of sodium ions and water take place in DCT. Thus, it maintains the pH and sodium potassium level in the blood cells. Next, move on collecting duct. It is also called as duct of Bellany. Collecting duct is a long straight tube where H plus and K plus ions are secreted to maintain the electrolyte balance of the blood. This is also the region where the maximum reabsorption of water takes place to produce concentrated urine. Collecting duct is also called as duct of Bellany. Collecting duct is a long straight tube where H plus and K plus ions are secreted to maintain the electrolyte balance of the blood. This is also the region where the maximum reabsorption of water takes place to produce concentrated urine. Functions of nephron. It converts blood into the urine, reabsorption, secretion and excretion of numerous substances. To remove all waste products including the solid waste and other excess water from the blood. As the blood passes through the glomerulus with high pressure, the small molecules are moved into the glomerular capsules and travel through a series of tubules. The cell present in each tube absorb different molecules excluding the glucose, water and other beneficial molecules. Today class is over. See you next class. Thank you.